So you are mostly water, 70% water, 30% chemicals, and the most common chemical that you're made out of is protein at 15%, and RNA, inorganic molecules, and so on to lesser degrees. So those macromolecules are going to be made out of those subunits, like I said. Sugars are polysaccharides are made up out of sugars, proteins are made up out of amino acids, and nucleic acids are made up out of nucleotides. Polysaccharides are held together by glycosidic linkages. Proteins are held together by peptide bonds, and nucleic acids by phosphodiester bonds. But in each case, we're going to use a dehydration synthesis reaction to link together the subunits into the polymer. So, so dehydration synthesis reactions require the input of energy, so they are endergonic, have a positive delta G, and so are non-spontaneous. We can break these bonds in in a hydrolysis reaction. So hydrolysis reactions are catabolic reactions. They are spontaneous, give out energy, and so are going to have a negative delta G. So we're going to talk a little bit about chemistry just to review. Anybody who has had chemistry might remember that we can get a lot of diversity of chemicals by having more carbons, by having branches, by having the presence or absence of rings, presence or absence of branches, more. You can always add another methyl group. And you can also add any of the functional groups. For instance, you could add a hydroxyl group, or you could add a aldehyde or ketone. You could add a carboxylic acid, esters. You can add an amino group. Um, the carboxylic acid is going to be ionizable, and so can give up that hydrogen and um, become negatively charged. And the amino group is going to act as a base. It can take on another hydrogen um, and become positively charged. Um, so another important group that is um, going to happen is these nitrogenous bases in rings. So um, so aminos can be added, or also nitrogens inside of rings. These are going to be important for hydrogen bonding. Um, we also want to remember the functional group phosphate because of the ATP and the energy associated with the trinucleotides for polymerizing DNA and RNA. I want to remind you that that intermolecular bond, the hydrogen bond, um, is between um, the partial positive on the hydrogen and the partial negative on the oxygen between two water molecules. It's a weak intermolecular bond, but it's going to be important in life. And it's really important in how water acts, and how water acts has really dictated how life. So what I mean by that is the interaction of water, one water molecule with another, is responsible for adhesion and cohesion. That's how we're going to get water to go up through the roots and up to the leaves through that xylem. It's also going to create surface tension, which will allow, like, say, a water strider to glide across the top of water without falling through, because he doesn't break the surface tension. So that's all due to those hydrogen bonds. Another thing that hydrogen bonds are responsible for is the high specific heat of water and the high heat of vaporization. So that allows you to cool yourself off when you sweat. Um, Another thing that hydrogen bonds are responsible for is the three-dimensional structure of ice crystals. take up more space because of those hydrogen bonds than the water that they were made out of. And so water is less dense when it's solid than when it's liquid. There are only two um, molecules that behave like this, ammonia and water. And so this is important for life because ice floating allows for liquid water to stay at the bottom of the lake, and that liquid water allows for the animals within the lake to stay alive, even though the lake is frozen over. So um, another thing that's going to be important about water being a polar molecule is that it acts as a great solvent, especially for ionic substances like sodium and chloride. The only thing that was holding the salt together was the the attraction of the negative on the chlorine to the positive on the sodium. So if you can balance out that positive with the negatives on the oxygen, then the sodium will just go into solution. So water is a really good solvent of all polar things, sugar and salt, but it's not a good solvent of non-polar things, hydrophobic things, things like fats. The more hydrocarbons you have in a chain, the more fatty or greasy it's going to behave, the less soluble in water it's going to be. The only way that we're going to be able to get it out of solution, get it into solution if it's really hydrophobic, is to put an amphipathic molecule in there, and we're going to talk about that later. So um, we want to briefly touch on acids and bases. An acid is anything that will increase the hydrogen concentration of the solution, and a base is anything that will lower the hydrogen concentration of the solution. So carboxylic acids work as a weak acid. It'll depend on the pKa of the molecule, what pH it will give up hydrogen. Um, so in life, we try to keep most solutions in a just barely basic, um, maybe in the 6.8 to 8.5 region. Um, acidosis in the blood will end up killing you. And so we have several buffers in our blood to maintain our blood pH. Um, there are situations in the body that are very acidic, like, say, stomach acid or inside of a lysosome. But those are highly compartmentalized um, uh, situations. We go to great lengths to keep the 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 acid sequestered away from things that acid would damage. Um, so uh, so uh, I also want to talk a little bit about different sugars. So there are monosaccharides, and they can be trioses, pentoses, or hexoses. Probably the one you're most familiar with is glucose or fructose. Those are both hexoses. They are C6H12O6. Um, then they can bounce back and forth from their ring form to their linear form. Um, um, there are many isomers for sugars. For instance, glucose has several isomers like galactose and mannose. They can be linked up to each other, either alpha or beta. So if they are linked up alpha, we can use them as a food source. Think glycogen or starch. If they're hooked up beta, uh, you cannot digest them. No animal on the planet can digest them. No eukaryotic cell can digest them. And so those are the celluloses and the chitin. They cannot be used as an energy source because we cannot break the when they are hooked up beta. So the sugars can be monosaccharides or they can be disaccharides, like say sucrose, which is a glucose and a fructose hooked up alpha with each other, and then they can be oligosaccharides. Oligosaccharides are things like starch, and glycogen are the alpha polysaccharides, and cellulose and chitin are the beta polysaccharides. They can be branched. They can get very complex. Um, then there are how fatty acids and other lipids hook up with each other. Like I said before, they can be either um, saturated or unsaturated, and when they're unsaturated, they're bent, and when they are bent, they are less dense, and we usually see them in fats and oils as triglycerides. That is three fatty acids hooked up to a glycerol molecule. Um, in the phospholipid bilayer, we have two fatty acids that are attached to a phosphate, and then they also have a head group. The phosphate and head group are polar, and the fatty acids are nonpolar. We're going to talk more later about the different head groups. In this case, it's choline. So we will talk more about lipids later. Another kind of lipid is the steroids. The steroids are things like the cholesterols, which are used in your cell membranes for rigidity, and the steroid hormones like testosterone, estrogen, progesterone, and some of the stress hormones like the glucocorticoids. Then there are amino acids. Like I said, they have different R groups on them. They could be basic, or they could be acidic. They could be non-polar, or they could be polar, uncharged, or they could be polar charged, like the acidic and basic. And then, like I said, the DNA, you've got a purine like guanine and adenine, or a pyrimidine like thymine, cytosine, and uracil. And this nitrogenous base is hooked up to a ribose and then to three phosphates. The ribose can be either ribose or deoxyribose, where one of the OHs is gone. They can hook up to each other to create DNA or RNA, respectively. So this is it for macromolecules. Try to review these three videos until you really understand what the different monomers are, what the different polymers are, how they're hooked up to each other, some of the uses of fats, proteins, nucleic acids, and sugars, and be ready for the quiz.